as excuse me. As Vivian said, we're going to talk about our latest OncoLand updates. So it's actually the 2015 fourth quarter update. And as most of you on the line have seen this before, you know the drill. I'll walk you through the updates in the slides and then jump back and forth between the software. I think the majority of what we're going to talk about today is actually is all data related. So in terms of features, I think it's just going to be mostly the new data. But there's some pretty exciting stuff that we're doing in this update, and that we'll be doing in future updates as well. All right. So I think the biggest part of the OncoLand update this quarter is updates to our OncoGEO 2015 data sets. If you guys remember, this was in OncoLand, which I switched to here. This had some RNA-Seq data in it that was in GEO and the sequence read archive. And I have mentioned to some of you that I've talked to that it was going to be the future home of holding expression data for GEO, and so that's what it's going to be. It's also going to include a new data type for OncoLand called comparison data. So this was previously in ImmunoLand and CVMLand, but it was requested by a number of our users to put into OncoLand as well. I'll explain what a comparison is in a minute, but in this update we have almost 300 comparisons across 87 different GEO projects, and this is going to grow each quarter. If you have areas of interest or specific, excuse me, if there's any areas of interest for these data sets, please send it to us. And also, if there's a missing GEO data set that you want, you can certainly send that to us as well, and we'll try to incorporate it in the next release. The big thing, at least for me when I'm working with this data, is that it's classified into different categories. And you'll see that here, disease versus normal, treated versus control, disease 1 versus disease 2. And so a lot of the visualizations are going to be based on those categories. But we've also curated the data fully, so I'll show you that as well. So let's take a look at the software. This is OncoGEO as you're used to, and this is your sample distribution as you're used to. By default, we're grouping them by disease or disease category sometimes, and then you'll get a more specific disease level that you can look at as well. You can also get tumor type, like you had. I think it was tumor type. Well, anyway, we'll show this in a second. Obviously, everything is interactive, so selecting points will show you the samples down below. And then what you'll notice is that we've done all the curation, so you'll see things like disease state. I guess that's what I was looking for. So these are all the individual disease states that are covered in this land. Cell type, so every single sample has had a cell type associated with it, or sample source. So it's basically a tissue or a cell type. You should see that here, sample source. This is going to be the tissue or cell type, which is pretty important for filtering. So if there's a particular cell type you're looking for, you can come in here and look at it. We're continuing to add RNA-Seq data into the land as well, so that's all in here as well. And then, of course, we still have the land tissue column that we've used in the past, and we're classifying samples as either tumor or not. I think everything in this land is going to be classified as a tumor, although there are some normal controls as well. One thing that you'll notice in selecting the views at the top level is that there is now a new comparisons view here, and you can see it says 4,500 samples in this land, and then 294 comparisons. Just to point out the data types, there's about 1,200 RNA-Seq samples in here, and then about 3,000 expression samples. So that could be Affy arrays, it could be Agilent Illumina arrays, but they're all expression related. So, if we click on comparisons here, we can take a look at all of the comparisons, and this is showing you the comparisons at the different tissues. So if we were interested in the treated versus control comparisons in breast cancer, I can select that. You have about 45 different comparisons, and we can look at all the curation that was done here. The comparison contrast, if you've never seen one of these comparisons before, is an important column because it'll tell you what was actually compared. So for instance, this one, this GSE 16179, it was a treatment, and it was comparing different treatments back to DMSO, whereas this one was treatment, estrogen versus vehicle control. And you can see, as I'm selecting the different types, like if I go in and select the disease versus normal types, you'll see the comparisons are related to disease instead. So primary tumor versus normal. And so what we've done is we've actually run an ANOVA or the correct model on all of these different comparisons. You'll see some of the other controlled vocabulary here. I mentioned tissue. Obviously, all of these are filterable. What sample IDs went into the comparisons, you can tell if any of the samples were actually removed. And then we try to control for treatment and tissue source or sample source, and a few other categories as well. Right now, everything is human in this land, although I think in the future we'll expand organisms, just as we have for Immunoland and CVM land. For individual projects, we've actually also incorporated the clinical data, and I'll show that a little later as well. So this land works like every other land that you've worked with, in that you type in a gene of interest or multiple genes of interest, select it, and search for it. The big difference I think that you'll see when you do the search is you're going to see a different visualization than you're used to seeing. For most of you that have worked with our data before, you're probably used to seeing the gene FPKM or one of the mutation visualizations as being the standard visualization that you see. So something like this, where it's controlled by grouping and it's showing you your different disease categories. But in OncoGEO land, what you'll see is this visualization instead, and I'll explain this. So what we have here is on the y-axis, we're showing our different disease categories. You can see them up and down here. On the x-axis is going to be the fold change, the log2 fold change. And then each one of these dots is actually an individual comparison, comparing disease versus normal. So all of these dots are the disease versus normal comparisons that are actually contained in this land. We're colored by sample source, so you'll get an idea of the tissue types. So you can see, in the gastrointestinal system cancer, you get a couple different tissue types. Same thing for reproductive organ cancer. And then the size of the p-value is indicative of how significant it is in comparison to everything else here. So, a tiny little dot like this is less significant than a larger dot like one of these. And I put that into the slide just so you guys can understand all that as well. Obviously, everything in our system is customizable, so if you want to change the groupings and stuff, you still have the ability to use the task tab, and you can also create custom views that get into a little more detail. At this point, I think the disease category is good enough for the y-axis, but you have the potential to change this to something like disease state instead, and get a little more detailed. But then you can see it kind of spreads out. So that's why we use disease category as the default column here. Okay. Data selection, like everything else in the system, selecting a point will show you more details about that selection. So selecting this point here is actually going to give me a plot of the original data. So it takes me back to the original data, and you can see very clearly this gene is downregulated as we expect in tumor versus normal. Changing dots will change the selection, so this one is upregulated, and selecting multiple points gives you the ability to scroll, as you see as I'm scrolling here, through the different data sets. You also have the ability to look at additional details about each of the selected comparisons. So if you click this plus button, I know a lot of people find this to be hidden, even though we made the plus bigger. You'll see different levels here. So here's the gene level, and so now you'll see all the details about these comparisons that are similar to what I showed you in that high-level comparison tab. So it's showing you all the information. One useful column to look at is the sample size cutoff. Just making sure that there's enough power behind whatever study you're looking at. So you can see some of these are pretty big studies. So each group has a large number of comparisons in it. And then everything's summarized at the gene level, so you're seeing a full change and a p-value at the gene level. But you can get back to the probe set level as well. So there's going to be individual probe sets, and there might be multiple probe sets per comparison. Okay. Someone asked if the axis can be changed to full change instead of log2 full change. Yeah, you could just change the transformation and do a signed, I think, exp2. So it's something like that. It adds the open space in the middle. Okay. This is the one type, but we also have other different categories. So as I mentioned, we've classified all of our comparisons into things like disease versus normal. Disease 1 versus disease 2 would be something where you're just comparing two diseases, so it's not necessarily going back to normal, but it could be, one type of breast cancer to another type of breast cancer. Sorry, if you hear the dog barking, that's the downside of working at home. So the next one I wanted to show you is treated versus control. And so we've classified all of these different treatments, and we have the same exact plot. So you're seeing, these 3 dots here are all from the same study, and it looks like we're comparing at different time points the treatment with lapatinib, I believe. We scroll over, we'll see the individual treatment. Treatment none. Anyway, that's the general idea here. Maybe this is treatment none. Zoom in here. Looks to be TGF beta is what these are treated with. All right, let's fix the zoom here. We can also do all comparisons. I kind of like this view. So it merges things. So you have the disease type first, and then what the treatments are. So as this database gets more and more filled, it could potentially become a little more useful. But you'll see things where it's up with disease and down with treatment, or vice versa. Just to show you how this, you can customize this further. What I did is I actually took tissue and I added it to this plot, and then I filtered for just my tissues of interest. So something like this, where we take tissue. So now we have tissue, and then we have disease, and then we have the treatment. And it's a lot to look at once, so you might just come in and say, okay, I just want, lung, liver, breast, and brain. And actually, you have to do that after the filter. And you can see now we can kind of zoom in, and you can say, okay, well these are clearly up, and then what's going on, with this one's down, and you can make your decision like that. Okay. As you're looking at these comparisons, you might decide, okay, well, this is interesting. I'm just going to reset my filters. and go back to the disease category, disease versus normal. You might find a bunch of breast cancer comparisons, for instance, that are of interest. So both of the genes down in both of these. You now have the ability to then say, okay, show me the rest of the information about these comparisons. So click Browse Selected Comparisons. And it's going to take me to the data underlying the full comparison. So by default, we're going to show you some Volcano plots, and you can then interrogate this data. So like the same way you would in a normal Volcano plot. Here's in the here's primary tumor versus normal in this comparison, and here's something similar in this comparison as well. And interestingly, these three genes are not really changing in this experiment. You have a whole set of different filters and visualizations here. The Volcano plot's the first one, but you can get to just a table if you want to export it of the p-values and fold changes. Everybody always asks about the missing data. It's going to show you all of the genes in the gene model, but not all the genes will necessarily be on every chip. So some of the genes are you're going to have missing data by default. But you can obviously then export this table. It's just, for each comparison, I have full change, raw p-value, the adjusted FDR p-value, and then repeats for the next comparisons. There's a Venn diagram view that I find handy. So you can look at, what's overlapping between your two experiments. So then you get a list of the 547 genes that are overlapping. You might say, okay, yeah, but what about the cutoffs? Like, what cutoffs are you using? This is just using default cutoffs of 0.05 and no fold change. So you can actually change greater than 2 or less than equal to 2 fold change and a p-value of 0.05. But if you want to make it more stringent, you can increase the fold change, and now we're down to 10 genes. Or you might increase the p-value cutoff or use an adjusted p-value cutoff instead. There is a view called significant genes that I like. It will show you how many, for each of your comparisons, it'll show you an up or down arrow of the genes changing. And so if you have like 10 comparisons, you can sort this and say, okay, just show me the ones where it's down in all 10, and then maybe export that. You also have the ability to filter by gene. So if you have a list of genes, you can import that list here, and then there's a pathway filter that's based on the molecular signatures database that you can filter by as well, and that'll filter some of these different visualizations. Comparison annotation just takes you to the full annotation about those comparisons, same as we saw before. And there's a few others here that could be useful. Summary statistics, another one that just gives you kind of an overview of each of the experiments. How many genes are there in the experiment? What's the most upregulated gene? How many genes are changing at different p-value and fold change levels? Just so you can get a kind of a general idea about the experiments themselves. You can always download these individual experiments back to Array Studio as well, using the download commands inside the download menu here. So if you wanted to like fully reanalyze everything, you'd be able to do that. But generally, you can work with it from within the lands. Everything I've showed you so far for a single gene or a gene set has been based on comparisons. We have not forgot about the underlying data. As you can see, you do have the ability when selecting something to show the profile down below. But what if you wanted to see all the individual projects or subset of them and what they look like? We have in both the expression category and the RNA-Seq category, we have a project view. So if you click project view, it'll take you to all the projects that you're filtered for, and it'll show you the box plots automatically. So if there's a particular project you're interested in, you could type in the author or the GSE ID, and it'll filter and just take you to those projects. The box plot's going to try to be smart, so it's going to use some basic calculations to figure out which columns should be used for box plots. So not all the projects will necessarily show a box plot, but most of them will. So it's kind of it's automatically doing what you might do in Array Studio normally in terms of creating these variable views. And obviously we're not limited to just a single gene. This all works with multiple genes as well. From any of the expression or RNA-Seq level visualizations, selecting points will show you all the details, including the expression values. But then you also have the ability to click clinical details, and that's going to show you all the other annotation associated with a particular geo project. So you're not going to just be limited to cell type and disease state and sample source that are part of the main metadata. We're going to include all of the metadata that was actually included in a particular experiment, and that will change, depending on what experiment you're looking at. So this one has tumor size, previous chemotherapy, that type of thing, histology. So everything has been incorporated, and we've tried to clean it up and use controlled columns as much as possible. Okay, so that covers OncoGEO 2015. I really think it's going to be a highlight of the system, and it's going to grow a lot. So if there are individual projects you want added, or types of projects you want categorized or added, I would say just let us know. We'll do our best to incorporate it. Okay, so on to TCGA updates. The main updates for TCGA are adding all of our clinical data has been updated as it usually is. The CNVCall, which I'll explain in a minute, data has been updated as well. We have about 1,600 new somatic mutation data samples. We've updated the marker paper in the clinical data. And this is something I'll go into more detail because I'm not sure that the people are using it as much as they could. We've also done HLA typing, which I'll explain in a few minutes as well. There was a question going back before I get to that to the GEO data sets. The comparisons are not based on what was already calculated and put into GEO. They're calculated by us. So we've gone through and curated and looked at the samples and then also created the comparisons. Okay. So on to TCGA. CNVCall data has been updated. And so what this is, if we are in TCGA here, just as a reminder, when you search a gene, we'll return different visualizations. By default, the amplifications and deletions are based on GISTIC2. So that's what we mean by CNVCall. TCGA releases the GISTIC-call results. So if we look at the CNV tab here, you'll see that things are called amplified, deleted, gain, heterozygous deletion, and missing. And you have the ability to change this depending on if you want to use the GISTIC2 calls where it's already been decided, or you want to use copy number cutoffs to decide. And it will change the number of samples that are amplified or deleted. And when you switch to copy number cutoffs, then you have the ability to change segment size and decide what's actually considered an amplification or deletion. Whereas for GISTIC2, it's already been pre-classified. So that's all been updated. And just as a reminder, this is also available under the copy number tab under summary. So this will just be This will just be the copy number data without the mutation data. Same filters apply. DNA somatic mutation updates of about, I think it was 1,600 new samples. So you'll see now when doing a search, the DNA alteration distribution will have probably more samples than it did before. And as a reminder, under DNA-Seq, you always have the DNA-specific views. So the somatic mutation distribution here, which is, again, we just have more samples than we did before. There's also the genome browser, which you can always look at, which looks at where the mutations are on the gene itself, and then groups it by whatever category you're using. And then I also like Somatic Mutation Landscape, which will show you, the most frequent mutations. So you can select this one to find out what samples they are, and what the actual mutation is. And whether it's a known SNP or unknown SNP or what have you. Okay, one thing we did a couple releases ago that I wasn't sure if people were using or not was start adding in the marker paper data, and I'll explain what I mean by that in a minute. But these are the papers that we've added in the last release, and so this information is included in the clinical data. So we've categorized it by marker papers, I think is the category. And what the request was from the user, and I think it was a good request, was that a lot of these TCGA papers, there's going to be clusters and categorizations that were done to the data in the paper. And so, previously you had to kind of import that yourself. You had to create a sample set, include it. So we've just gone ahead and included that for you, and you can use that in all the different visualizations and analytics. So here is there's actually a category called TCGA marker paper, and you can see we've categorized it by different types of papers and columns. And so I'll show you this live here. So we'll go back to TCGA. So if we just type in marker in the search bar here, what you'll see is all of the marker columns. So, if we looked under mRNA, you're going to get the PAM50 columns from the breast cancer papers. There's different clusters. There's cluster of clusters that they like to do in some of these, different pathway classifications. So if you're reading a paper on the data, you can use this. So for instance, this lung adenocarcinoma pathway, I would be able to just type in that, and then we could then group and see how many samples there are in the different categories. And you can see that this is not a highly, there's only about 239 samples that have this classification. Although most of these missing are probably not lung. So the example I was going to show was in kidney. So let's just filter our data for KIRP. It's the kidney renal I'm not going to try to remember the name of the disease. Kidney renal papillary cell carcinoma. And so there's a marker paper about this, where they've basically classified the samples into type 1 papillary RCC and type 2 papillary RCC. And so we can use that in our different visualizations. So there's a pathway in the paper that they mentioned, the NRF-ARE pathway, NRF2, I believe. So it's this one here. So I'm just searching for the pathway. And in the paper, they said that they saw more mutations in this pathway in the type 2 classified samples than they did the type 1. So we're just going to check to see if that's true, and then see if there's any new samples that you might be able to classify based on that. So what I'll do then is I'll filter for just the KIRP samples, and then I'll change my grouping here to the column, the PRCC subtype column. And what you see kind of confirms that there's about 8 samples that have alterations in these genes in type 2 and only 1 in type 1. And then you're actually seeing a whole other set of samples that also have alterations in these genes that were probably samples that came after the paper was written. And so you might be able to then classify these as type 2. As well, and then of course you can look at the expression data. You can look at any of the different visualizations using this this column. And again, if there are papers that we haven't included that you'd like us to include, please let us know. So this is just your standard RNA-Seq expression patterns for these genes. Okay. The next thing I wanted to mention was updates to the TumorMutation Land. Just as a review, this is a land that contains publicly available mutation data covering many different disease types. So essentially, if a paper is written that just releases the mutation data, we're going to try to include it here. The source column links back to the PubMed article if you want to try to find that. And we now have about 48 total data sets in here. I say mostly mutation. If they released copy number or expression data with the same paper, we'll try to include it here as well. So there is a bit of copy number and expression data in here. But just to review, this one, here's the TumorMutation Land. We have a disease column now, so you can kind of see all the different diseases that are covered here. And then, like everything else, it's all gene or gene set based for searching. And so you'll be able to, you can look at the mutation type views across all these different data sets. So really, the key thing here is just the fact that we have more data than we had before, more data sets. All right, last big update, which I think is kind of exciting. We've had some requests by, oh, there's a question coming in before I move on. Did all the mutation studies contain matched normal? I don't know the answer to that, but I would assume that if it's been classified as a somatic mutation, that it would have matched normal, or would have been called by that. Called to have matched normal. As far as us having the matched normal in the lands, we wouldn't have that because we're just going to have the mutations themselves. Okay. We've had a number of requests from our users to use the Optitype or there were a couple other algorithms that were suggested as well for typing HLA class 1 or potentially class 2, but we started with class 1 because that's what Optitype supports for all of the samples in our lands. We've started doing this with both the CCLE and the TCGA data sets. And the TCGA, obviously there's 11,000 RNA-Seq samples, so it was a pretty big undertaking. The idea here is that you can easily search these data sets and use the class 1 types as part of your different visualizations and filtering and analysis. For the cell lines, there's been a number of papers, and I listed this one here for you guys to read on how this could be useful. But if, for instance, you needed a breast cell line that had a particular HLA-A type, you would be able to easily filter and see that here. And I'll show you that in a second. And then for TCGA, some example things that you could do with it would be to check population frequencies of HLA types to ensure that there's expected distribution. You could potentially identify subsets of patients with particular signatures for further follow-up. And then, you can also do things like if you found a target gene where you're seeing an overexpression, you want to ensure that it's not, that every sample that's upregulated isn't a particular HLA type. It probably wouldn't be, but these are the type of questions that I've been told people would potentially use this for. So taking a look at this, this is CCLE. I built some custom views for this. These aren't included by default, but if you're interested in including this in your lands, I thought they were kind of useful. It's just at the top level of CCLE. I have HLA-A, B, and C sample distribution views. So it just gives you, and you can obviously customize this, but it gives you for each tissue type the different HLA types there are for that tissue type. So if you wanted to see for breast cancer what tissues have O201 as your type, then you would see, okay, it's these 9 cell lines here. And then in CCLE, we've included this all in the main metadata, so you'll see the types here. The concept is you have 2 alleles, so you're going to get 2 different calls. So this one has A201 and A1101. And then what we did is we merged them together in like a main column, so you could do some nicer things for filtering. And then so you'll get that for the HLA-A, B, and C. These are obviously available in our filters as well. So if the only thing you cared about was looking at sample details and saying, okay, show me any sample that has A201, you could filter for that, and it'll show you the 291 overall tissues, or sorry, cell lines that have A201. So I think I've been told that the cell line stuff is going to be pretty useful for everybody doing this type of work. And then the follow-up to that was doing the same thing in TCGA. So once again, I have created a custom view, which is not going to be necessarily available by default to everybody, but if you like it, feel free to ask us and we'll add it. And so for each HLA type, you once again get a sample distribution. And so this will give you, for each tumor type, what the distribution is. And of course, these columns can be used in any of your groupings and any of your different visualizations. But so if you were looking for, actually, normally what I do is I get rid of we only look at RNA-Seq. It kind of cleans this up a little bit. Oh, there we go. If you're looking for a particular HLA type, or you want to create a subset of a particular HLA type so that you can do different comparisons, you could do that from here. If you have questions about this, feel free to shoot our support team an email or shoot me an email. But it's, I think, something that we're going to continue to do for the other lands as well. And then the final things that I'm not going to show anything before we go. We've added some additional RNA-Seq samples into the blueprint data set, and then about 145 RNA-Seq samples into the ICGC data sets as well. Last thing before we go is a preview for the next update. The next update will be announced at our user meeting in Cambridge, and then we'll for those of you that can't attend, we'll post a video after that, but it'll be it'll be one of the talks. The biggest news, well, there's a couple of big pieces of news here. One is that we'll be updating all of our major lands to B38. You'll see that we're still going to be updating the B37.3 for the near future. So we're starting with TCGA, GTEx, and the Cancer Cell Line Encyclopedia. So you'll basically get, for each one of those, you're going to have a B38 version and a B37.3 version. We're moving to the GenCode gene model, the basic version, for visualization and searching. The UCSC gene model was phased out, and so they're using Ensemble or GenCode. So we're going to do the same thing. And so I think that's going to be a big improvement for people because it'll match up with a lot more of what's in literature when you're doing your searches. And then the other huge piece of news is that we are going to process all the DNA-Seq samples with GATK and provide results for that. So we're going to do that ourselves. So that's going to be the germline data. That's going to require controlled access. If you're interested in this, you should contact us, and we're going to have to go through a process of applying the TCGA together so that we can share this information. And then finally, we'll be updating OncoGEO 2015 with some more comparisons and more data in the future releases as well. And I'm sure there'll be other updates as well, but these are the highlights. So thanks everybody for attending. If you have any questions, we'll hang out for a minute or two for you to ask. And then as Vivian mentioned, she's going to post the make the slides available as well as the video. I did put, if anybody can figure out how to use it, there's a handout tab in the GoToWebinar thing, and I did put the slides from today in the little handout tab. I don't know exactly how you download them, but it is there. But if not, we are going to post them, but we'll send that out to everybody.
Hi, Matt. There are a couple questions coming in. Could you see the questions, or do you need me to actually get it out for you?
I think I've answered all of them except for this last one. Will all the data be available through text dumps? And is there an ETA through the text dumps? Yeah, we'll make everything that we're processing will be available through the text dumps. Usually it's 2 to 3 weeks after we actually released the lands. So we'll give it 30 more seconds, and then if you do have other follow-up questions that you didn't ask, just shoot us an email, support@omicsoft.com, or you can email me directly. As a plug, if anybody has ImmunoLand or CVM Land or interest in either, we're going to be having a webinar at the same time tomorrow, and I'll go through the updates to both of those lands as well. So with that, I think I'll stop recording, and hope everybody has a nice day.
All right. Thanks, everyone, for your attendance. And please also check out our blog and recorded webinar on our website, omicsoft.com, for the latest news and updates. So thanks, everybody, and hopefully we'll see you next time soon.